So that was a very interesting discussion on crossover designs and how to analyze crossover trials. You have to worry about the period effects. You have to worry about the sequence or carryover effects. But one interesting thing is that most people do not do crossover trials because they're difficult to do. They're not possible in multiple situations. And so while it's interesting from a statistical standpoint, we don't often do them. One thing that we always do is also interesting from a statistical standpoint, and that is the need to do interim analysis. And interim analysis only means analysis that you do during the trial. You don't have the luxury to wait until the end of the trial to analyze your data. When you do an observational study, you have that luxury. You collect all the data, you clean all that data, and then you do the analysis. But in an experiment, especially when the experiment is on a health condition that is affecting people, we already heard 2 days ago that we can only do this when there is equipoise, when the interventions are not known to be one better than the other. That's the only reason you can even contemplate doing a trial. If one is known to be better, then it's unethical to do a trial because you already know that one is better, so therefore the other one is inferior. So you can't do it. You can only do it when there is equipoise. Well, if your study is a short duration study, what if it's a surgical technique that will last 2-3 hours of the operation, and the outcome is known in the first 7 days? You're able to get a lot of patients. The necessary sample size is whatever it is, and you're able to recruit them, and you'll know that result in 2 months or 3 months. There is no time to do interim analysis. Nobody does interim analysis. So it's not a required thing to do unless it is a long-term trial that because you may be already off equipoise in the middle of the trial, you need to then potentially stop the study and let the results known, because then you'll have to change what you're doing to the control arm if it turns out to be effective. And you would also help out people with that condition that are not in the trial in your country and worldwide. Okay, so the duration of the trial is one, the type of intervention, the type of condition that you're studying demands that you do it. If you're doing an educational campaign or a behavioral intervention of diet change, for example, do you need a data safety monitoring board? Do you need to do interim analysis? Possibly not. If you're doing an intervention that is a exercise one, possibly not, but possibly yes. Why? Because there's adverse events. We did a study of adverse events a study that was looking at obese people and changing diet, but there was also this recommendation you should be a little bit more physically active. One person decided to run a marathon, had never run a marathon, had a heart attack and died. Then, a serious unsuspected adverse event. This trial was almost stopped for this one crazy person who decided to do this. So you have to have somebody monitor. Most studies in North America, the government forces you to have a data safety monitoring board, like Kumar mentioned yesterday. But interim analysis may be necessary, especially if it's you have a lot of potential safety or adverse event issues, or if there is a potential long duration, and if efficacy is shown before you may need to stop early. So what I'm going to talk about is when do we require interim analysis? What are the complications of doing interim analysis? And how is it done from a statistical standpoint? Okay. So we'll talk mainly about group sequential methodology. I'll mention some others that exist, like stochastic curtailment and conditional power. Those are more Bayesian approaches, our Bayesian friend over here. We'll talk about that a little bit. And what happens if we stop early? People say, oh, stop the study early, the results are divulged. There's a whole lot other things that happen when a study is stopped early, and I'll talk about that in an example. So, some examples of studies that have been stopped early. They can be stopped early for safety reasons. You have either a higher rate of serious adverse events than expected overall, or in one arm more than the other. And we call it, this is a study on intensive insulin therapy and pentastarch with people with sepsis. I mean, this is very tough condition, and you have a lot. They had a rate of severe hypoglycemia was observed, and the rate of serious adverse events was very different between the groups, 10.9% versus 5.2%. They haven't powered the study for this. So the Data Safety Monitoring Board is looking at the data, and if they think that those rates or the differential rates are unacceptable from an ethical standpoint, they can recommend to stop the study. This is beyond the control of you as the investigator having designed a study like that. That was bad luck that this was happening, that they had more serious adverse events in one group. This is a study in use of methotrexate plus UDCA for cirrhosis, primary biliary cirrhosis. It was stopped early for futility. And futility can be thought about different ways. Futility may be thought of, if I continue the study, when I reach the end, my event rate is so low that I won't have enough power to do it. So it's futile to continue. That's an efficacious lack of efficacy at the end. It also could be because I have been so sloppy in my data collection that I have so much variability, so much heterogeneity, that my big effect will be masked or hidden by all this noisy variance. And I've done a garbage trial, and the board may say, you're going to not reach significance at the end. Or it may be that, as one study that was done in AZT back in the early days of HIV, discovered in 81, 82, and the studies were, I mean, it was basically a sentence of death when you were told you had HIV. They were trying all sorts of things against placebo. And when AZT, I think it was, came out, they were testing this, AZT versus placebo. There was promise that this may work, belief that it would work. And at one of the clinical sites at University of California, San Francisco, a very enterprising chemist bought property right in front of the university and put in, we will analyze your drug to see if it's placebo or active. Pay us $500, we will do it for you. So the patients randomized would come, pay $500, and find out, oh, placebo. They invalidated the study. Futile to do it. It didn't happen everywhere, so they were able to test it someplace else. But at that site, it was, people do this. All over the world, people do this kind of stuff. Be careful. But you could have a poorly conducted study. You could have a low event rate because you were very optimistic in your design that you said, oh, the effect will be very large, and you're actually getting a very small effect. But the only ones that can look at this are the Data Safety Monitoring Board, and they can stop the study for futility. So they don't always stop the study for, they had no effect on the course of PBC or primary biliary cirrhosis treated with that. So they had very small effect, so they stopped it for that. That actually is a bad conclusion. Yeah. I mean, failure to show significance does not mean that you've accepted the null hypothesis. So this is a poorly reported one in that Journal of Hepatology. We mainly think about stopping a study early because the intervention is effective. But we also can, the study could be recommended to be stopped for other things. So here, this trial of atorvastatin in the CARD study was terminated 2 years earlier than expected because the early stop-in rule for efficacy had been met. So we will talk about where these things come about. So what are interim analyses? Interim analyses is a term that is often only used for the efficacy one, but there's a lot of other interim analysis that happen. There is internal interim analysis and external interim analysis. So the internal one is you or the coordinating center monitoring what Kumar talked about yesterday. There's all this constant monitoring of the accumulating data almost daily. You don't wait 2-3 months, oh, let me look at the data quality. You want to find mistakes early. You want to prevent them from occurring in other places. So you're almost on a daily basis. You have a study team that is the data manager and a bunch of other people that are looking at the data almost constantly. And you're looking at recruitment progress, you're looking at follow-up, compliance, the quality of the data. And then you may look at, usually we don't like it to happen, but some people do, is look at outcome data in the aggregate, overall, never by arm, what is happening with adverse events from a safety standpoint. Are we getting too many people that are having nausea overall, et cetera? But the Data Safety Monitoring Board has the privilege to look at that by intervention arm. So they look at the same study progress outcomes, but now by study arm, because maybe you have poor adherence in one arm versus the other. Maybe you have poor quality in one arm versus the other. So now the study integrity is compromised because of the performance of you as the investigating team. And they may stop the study for futility because of poorly conducted study, okay? It's not very common, but they have done that sometimes. More commonly, we think about them monitoring the outcome. They're monitoring the primary outcome mainly, but they also look at secondary outcome and subgroup analysis, et cetera. And they look at all of this by intervention arm. How do they do this? And what is the rationale? So there's ethics is basically the reason we do it. Without ethics, we would wait until the end because we've designed the study to be analyzed at the end, to have the adequate sample size at the end, to have the adequate power and number of events at the end. So why would we do it in the interim when we don't have the adequate event size? Because we are forced to do it by an ethics standard. The need to possibly early terminate the study if the study is being done improperly, study integrity is compromised, or if you already answered the primary hypothesis. So if you have maybe a study that is only 2 months, 2 months is not a long time. But if it's a year or more, especially chronic disease ones, we do 2, 3, 5-year studies. then there is a chance of something being answered before we get to the end. And so we are forced by ethics to look at it in the interim. Now we have considerations that are analytic considerations that may force us to stop. And that is the safety ones, unexpected or unanticipated toxicity, or unequivocal evidence of treatment difference in efficacy. So they are primary hypothesis, or zero tendency or unlikely possibility of a conclusive result, so that you get to the end and it's inconclusive. So why continue if you're not going to be able to answer the question? Okay, that's what people call futility, or one type of futility. But we also have administrative reasons that lead to the other type of futility. You have poor data quality, poor compliance, poor recruitment, etc., and therefore you have poor power. So they may say, you're not going to get anywhere at the end, so we'll stop you for futility for administrative reasons. Then there's other subjective reasons. You think that you are the only smart person studying this condition in the world. That's not true. If it's a condition worth studying, I can bet you that there are other people studying this. We were studying left ventricular dysfunction and doing the studies of left ventricular dysfunction using enalapril as the ACE inhibitor in a study in the United States, Canada, and Belgium. Meanwhile, in Europe, they were studying the same thing using captopril. And their results came out first. So an ACE inhibitor works. So why are you continuing your study? There's no need to study enalapril. Well, the investigators argued, no, but captopril has to be taken twice a day or 3 times a day. Enalapril is only once a day. So we want to, it's important to study it because people may use enalapril more because it's only once a day. We were doing that study on coronary primary prevention trial. I told you about drinking this horrible sawdust 6 times a day for 7 years. In the middle of that study, 2 or 3 years into it, captopril came out. Not captopril, Lipitor pill was invented. And somebody said, well, why are you continuing this study? No one is ever going to take cholestyramine. They're going to take Lipitor because it's convenient. It was not a horrible sawdust. Well, the investigators now argue, oh, no. It's not that we're testing the drug itself. We're testing the principle of lowering cholesterol. So what investigators will always argue, don't stop my study. Don't stop my study, because if you stop my study, the unused money that you have given my university, I will have to return. And I don't know anybody that likes to return money that has been given to them. So investigators always have a conflict of interest. They never should be involved in the monitoring. That's why Kumar mentioned the External Data Safety Monitoring Board is external to any of the institutions. They are not good friends with any of the investigators. They are not buddies or, classmates, batchmates, or whatever. They should be independent with no conflict of interest. So results from other trials may come out. Clinical practice may be different. And if it's a new drug, an existing drug that you're using, and you're studying it in renal disease, but it's also being used for GI disorders, and they're using it in a different group already on the market, and they're getting bad results with it for another thing, they may say that drug is not going to be used. They're going to take it out of the market. So why are you continuing your study? Those things, statistics can't help you in making that decision. I'm going to talk about the analytic things, because the analytic things are the things that statistics can help you with. So let's look at a hypothetical example. We saw that the primary analysis is a simple test. We saw that the test statistics are always can be thought of as the Z statistic, whether they're log rank or t-test or Mann-Whitney or Fisher's exact, etc., all the z-tests. We all know from our first course in statistics that the critical value, if you're doing a 0.05 level of significance, two-sided test, the critical value is minus 1.96 and plus 1.96. So if we have an example here where we have 5 looks at the data. We have at time 1, 2, 3, 4, and time 5 is the end of the study. But we're going to do 4 looks in the interim. What do we do? If we use the boundary of 1.96, what's going to happen? Hypothetically, we start the study, we start collecting data. At time T1, we tell the statistician, freeze the data, make sure it's clean. Oh, he's been cleaning it all along. The data coordinator people are cleaning it all along. Then they prepare the report. It takes about 2 months to prepare the report, usually. And they present it. And what you look at? The Z. Who looks at it? The Data Safety Monitoring Board. You as an investigator have no idea this is happening. The Data Safety Monitoring Board looks at it and says, we're now nowhere close to the boundary. So we tell the investigators, we met, in the minutes usually will say, we carefully examine all the relevant information, and our recommendation is that we do not see any need for the study to not proceed as per protocol. Bus, nothing else. Not Dr. X said this, Dr. Y said that, no details. So what happens then? The study continues. More data is collected. Now you get to time T2. You use all the data up to now, including what you use for Z1. And you calculate the next one. And pop, it's here. No trend. If the investigators knew this, they may quit. They may try to figure out how can I make the trend better and try to manipulate. Let's change the protocol. Let's do this. Let's do that. The Data Safety Monitoring Board says, we looked carefully at all the data presented to us, and we do not see any reason why the study should not proceed as per protocol. What happens? Study continues. You get to time T3, and you're here. I haven't crossed the boundary. So what does the study team know? Nothing. What does the Data Safety Monitoring Board tell them as feedback? We've carefully examined all the data presented to us, and we do not see why the study should not continue as per protocol. Same message. They don't say, oh, we are close to it. None of the stuff. Because if you know what is happening, you as an investigator, even though you tell me you will not, you will try to manipulate things to make things significant. So what happens? The study continues. More data gets accumulated, and you get to time T4. And what happens at time T4? The same, they prepare that, they present Z at time T4, and boom, it's here. What happens now? Well, you think, wait a second, we've crossed the 1.96, it's significant. We should stop the study. But there is a problem. What is the problem? Multiple testing. I've been testing it. I should adjust the alpha for multiple testing. And I don't do it with Bonferroni. Why can't I do it with Bonferroni? Bonferroni is for independent tests. These tests are so dependent because I don't do Z4 if Z3 was significant. So it depends on the value. And the data that I use for Z4 is all the data that I use for Z3, that I use for Z2, that I use for Z1. So the accumulating data makes these things so dependent. How do I adjust the alpha? Well, there's a little bit of history on that. First of all, I think it was Anderson in 1969. So when they started doing these long-term clinical trials, and the Bernard Greenberg report had come out that you need data safety monitoring boards. And now they were starting to look into it. Statisticians started to work on how do I actually adjust for it? If I do 5 repeated tests at the 5% level, Bonferroni would say 25% would be my type 1 error probability. But Anderson calculated it's not as bad as that because they're correlated. It's only 14%. But 14% is higher than 0.05. So nobody wanted that. They wanted to control it, to keep it at 0.05. So then they did sequential analysis methodology, incorporated the sequential analysis methodology into this. And sequential comes from the fact that the results at one depend on the results at a prior time. And data are analyzed repeatedly. In each analysis, you make this decision to stop the study or continue. And they started to adopt a sort of like repurpose methodology that existed from testing quality control in manufacturing production. These people doing light bulbs, they would do a batch this year, they would do a batch next year, they will do another batch next year with independent batches, and they would sequentially see whatever production quality, etc. They said, why don't we do that sequential production to see if we can do it? But we have to adapt it to the fact that we have accumulating data. And so the methodology was developed by Abraham Wald in 1947. He called it the sequential probability ratio test. And then Armitage and then Anderson did those calculations, and they did a truncated sequential design. If you're interested in that, I can give you the references. But these required that I do independent testing in batches of that. So we still had to repurpose it a little bit. So Armitage in 1969 is the one that basically we have to attribute all of this to. He called it repeated significance testing, and said that's where he got that 14.2% of alpha in his paper. And he said that if you tell me the number and timing of when you're doing the test, we can adjust correctly, okay? And that gave rise to what we call today group sequential designs. And so you have to have equal size groups of patients or of information, of events, are pre-specified number of looks. You're going to do 4 looks, you're going to do 2 looks, you're going to do 6 looks. And that gave rise to then ways of calculating boundaries that are different from the 1.96. One of those boundaries, well, so these design said you do K looks, and after each K look, you will calculate ZK at that look, and you'll have a critical value Z at that look. And if you are within that, lower than that boundary, you continue. If you're above the boundary, you stop. And this, for those of you statisticians, it comes from Armitage being able to, even though it's accumulating data, he was able to show I can get independent increments and use what's called martingale theory and Brownian motion to do this. Those of you that are not statisticians, you can forget that last sentence. But what he came up with is that now he enabled calculation of boundaries. One of the first boundaries to be proposed, this is now in the 1970s. So before 1970s, nobody was doing any of this. Stuart Pocock, the same guy that proposed recently the win ratio, he said, what we'll do is you tell me how many looks you're doing K, and we will then inflate the boundary by the number of looks. And he did some calculations, and he showed that if you're doing 5 looks, the boundary is 2.4. If you do 10 looks, its boundary is 2.5. So what it was is that now the boundaries, if you're using the proper ones, you cannot use the 1.96. If you do Pocock's, it becomes 2.4 whatever. And so it's blue. So now with that Z4 hasn't crossed that boundary. So what happens? What does the Data Safety Monitoring Board say? We have carefully looked at all the data presented to us, and we don't see any reason why the study should not proceed as per protocol. That's it. Then comes the end of the trial. And what happens at the end of the trial? The study is now ending. There is one final calculation of Z. The investigator is sitting in his office, biting fingernails, worried about the result because they don't know any of this thing that has been happening. They are anxious. They walk by the statistician's office. They look to see if he's smiling or not smiling. That's what Kumar says, that he goes and works at home. So nobody sees him, nobody looks at him because he may disclose something. So it's all secret. And what happens? Let's say Z5. So now the statistician goes to the investigator and this person is saying, okay, what happened? What happened? He says, well, sir, we got a Z value of 2.03, whatever. Yay, says investigator. It's greater than 1.96. It's significant. No. I have to tell you that we have been doing sequential testing, so we have to use 2.4, so it's not significant. Nobody's going to know this. We haven't registered the protocol. We haven't done that. This is what, 1977, there's no registration of anything. We don't have to tell anything to anybody. It's greater than 1.96, let's publish. But this is incorrect, right? This Pocock boundary is too stringent. 2.4 is big. So nobody uses it that much. Again, Pocock comes up with things that he himself doesn't like to use. So other people proposed, also in the 1970s, Hay-Bittle in the US and Pito, that same Richard Pito in the UK, proposed, you know what we should do is during the trial, make the boundary very, very high, 3 standard deviations. And at the end, we don't have to be so stringent as a 2.4. Those of you that work or have known about the 6 sigma, the people do quality control, they talk about 6 sigma. It's because 99.7% of the bell-shaped distribution is between plus or minus 3 sigmas or standard deviation. So that's the 6 sigma. Other than that, you're out of control. So he's come up with this also. Let's use 3. So it's simple, very easy to do. And so at the end, the penalty you pay is not from 1.96 to 2.4. It's just a tiny one to 1.99. Okay. So in that case, this happens. You would be 3. Oh, and that's Z5 is significant. Fantastic. We can publish, etc. So people started using the Hay-Bittle-Pito approach better than the Pocock. But then somebody started saying, wait a second, you're just doing this not for ethical reasons, wanting to stop the study early. When you're very close to the end, you not even have a chance to stop. You're doing 3 still. It's only at the end. All you're trying to do is save your alpha for the end. So people started criticizing the Hay-Bittle-Pito one. 1979, O'Brien and Fleming, working in the United States, came up with another approach. He said, conceptually, it makes total sense. Initially, when we are starting the study, we have fewer events. We're not very sure of the effect. I would not want to stop my study unless I have a huge effect. But as I get closer to the end, I have more information. I have more confidence in the estimate of the effect. And I'd be willing to stop early. And therefore, the boundary should be getting smaller and smaller. So the boundary is coming down and getting smaller. They had a complicated formula that depends on which look you're at and how many looks you're doing. And for them, at the end, they would be at 2.04, a little bit more than Hay-Bittle-Pito, but not so much badly above 1.96. So theirs looks like that. And I never met O'Brien, but I met Tom Fleming. He's alive. He's a very charismatic statistician. He was a member of most of the boards, and he convinced the boards to use his method. And the O'Brien-Fleming method is what most of the times you see is being used. Okay, and it's very attractive because now that Z5 would be that, you also had almost stopped with Z4, so you offer this, so it's intuitively very attractive to use. So all of these statisticians have PhD students, and Wang and Syadis worked on doing a generalization of some of these boundaries, and they came up with a much more complicated formula, and it's actually somewhere in between Pocock and O'Brien-Fleming, okay? And so for them, it looks different boundaries. So Pocock is constant. This is for 10 looks. It's constant over the looks. And so nobody likes it because it's too high here. O'Brien-Fleming is huge. You never stop here. But as you get closer, you allow the possibility of stopping. Hay-Biddle-Pito, you don't stop there, but you have very low here. And Wang-Syadis is somewhere between this and this. So it's not as high as O'Brien-Fleming, but it comes down. But it still is a little bit too high over here. Nobody uses that. Maybe the students of Wang and Syadis use it, but I haven't seen it. Now, at the Population Health Research Institute, we use a different one. But here, this is what these four look like schematically. Salim Youssef is another one of those physicians that doesn't like to stop a study early. But he said, we will allow a study to stop early, but we will restrict when we do the analysis. We don't have the liberty of doing analysis at all times. We will do it at specific time points. So he suggested that at approximately halfway through the study, don't do anything early, but halfway through the study, begin to look at it. And at halfway, you use not a Z of 3, you use a Z of 4, which is very restrictive, okay? But you consider stopping if you get greater than 4, and you would conduct another look within 90 days. Why did he suggest that? It's because when you cross a boundary, remember that data management is a big issue. It takes 2 months to prepare a report. So you are preparing a report with old data 2 months ago. And if your data management cleaning of the data is pathetic, then you may have to start it 4 or 5 months before. We try to do it, we do it 1 month before. You freeze the data to do it, but it's still 1 month old data. And the board members would like data as of today. Well, if the data gets frozen 2 months before or whatever, there's still data that's been accumulated, but it's not part of the report. And now you are examining it and you've crossed it. When you cross it and you stop the study, what happens? When you stop the study, that means that any new events after today are not included. Somebody may have an event tomorrow, it's not included. But if they had an event yesterday, they're going to be included. If you had an event that is being adjudicated, you have to include it. And if you have too many that are in that process of being cleaned and adjudicated, et cetera, if you just crossed it, it may be your bad luck that when they adjudicate these, it just uncrosses. And now you've stopped the study. When you've stopped the study, you can't restart it. You stop the study, you've told everybody, this is the result. Now the investigator is writing a paper on it. And then you say, wait a second, we added the other events and it's not significant. What? Come on. And how do I restart? I have to get my staff to start working again and get the patients to come in. We've already told the patients that we've stopped the study. It's impossible to restart the study. So he said, practically, if we cross the boundary, let's give some chance and do it within 2-3 months. Do it again. and see if we still cross it. So he does at around 90 days, because in cardiovascular disease, that's what he felt was good. And if it still is above that, then you stop or you continue. If you continue, you go to 75% of the information. And now you lower it to O'Brien, to Hay-Biddle-Peto's 3. Okay. So this is actually in the spirit of O'Brien Fleming. I have less information at 50% than I have at 75%. So I want to be coming down and getting to closer to the end. And they do the same thing. We'll do it 3. If we cross it, we do it within 90 days and do it again. And if we still cross it, we stop. If we don't, we go to the end. And at the end, we can use 1.96. So we don't have to use that 1.99. We don't have to use that 2.044. We can actually use the 1.96. It was very interesting because he proposed this in the year 2000 or so without doing any calculations. And I said to him 3 years back, we need to do the calculations. So I did the calculations. It's published in the Journal of Clinical Trials. It was accepted last year, but the paper actually came out March of this year. It's open access, you can get it. But what it looks like is this. This is actually in the paper. So you have now the Hay-Biddle-Peto. Sorry, not in the paper. No, in the paper, we just have a table. This is from a poster, but you will get this. So the Hay-Biddle-Peto is that this one here. This is the Pocock one, the O'Brien-Fleming one. And there's actually 2 PHI boundaries, one that has 4 here, and then 3, or has 4 here, and a stringent one of 3.5. But at the end, we come to 1.96. Okay. And if you're going 2-sided, you could do it this way. But you say, well, is this work? Yes, we actually did this calculations and simulations to show that the overall alpha for all the other ones is slightly more than 0.05, but this one is exactly 0.05. So we don't inflate the alpha with it. It's a very pragmatic boundary because it considers the reduction in uncertainty as you have increasing information, protects the overall alpha, as I said, and preserves a substantial amount of it, or almost all of it, for the end, so we can do the 1.96. And early stopping is possible, but you have to have substantial evidence that this is better, okay? An analysis at the end does not pay a high price for the interim looks, and it's not that complicated to calculate. The O'Brien-Fleming and Pocock ones are complicated to use. But the thing I like about it is it takes into account the pragmatic behavior of the board members. The board members don't like to do interim analysis early. So they never would do something O'Brien-Fleming. No, I'm not going to do that because you're just starting to study. You have 20, 30% of the information. Let's wait a little bit. So they rarely consider early stopping, only when you get in towards the end. And when they cross boundaries, they want to be very sure that we've got the real result. So they said, can you do the analysis in the next month or 2 months with the accumulated data to see if it's consistent? Are we still getting it? So that's where we got that idea. And when they get very close to the end, they tend to not like to stop the study. Because when you stop a study, what happens? You stop the study means, from a statistical standpoint, no more events are counted. But from a logistics standpoint, that's the nightmare. Suppose you're doing a study on 500 people, and the study is some active intervention versus a control, and outcomes are, so it's a rare, not a rare condition, it's a critical condition. people would be dying or having serious outcomes. And suppose that you um, you do the study and you get to the end. When you get to the end, what do you have to do? The investigators are thinking, I have to publish. I want to present at that conference in Hawaii because I want to go to Hawaii to present. But they forget what? They have 500 people that have volunteered part of their life to help you answer a research question. What are you going to tell them? If one of them has died, to the widow, you'll say, yes, the study was significant. I'm so sorry your husband got the control arm and died. Do you tell them to them in a WhatsApp message? Do you tell it to them or don't tell it and they read it in the newspaper? Is that ethical behavior? No. You want to bring them in. You have to carefully have some psychologists talk to them in a proper way. You have a script that you develop on how to tell people that we're under control, how you tell people that we're this, people that have that, what are the implications, what's going to happen in the future to your care. Are we going to care for you anymore? Are we going to say, no, you've done it already. Next time you come here to the clinic, you have to pay. We're not going to give you parking. We're going to treat you differently. You have to do something, and you have to plan what's called proper closeout procedures. When you stop a study early, if you haven't planned that, you now have to scramble and develop the closeout procedure to inform people. And people don't think about it. They only think about themselves as the investigators. They ignore the poor patients that have contributed to their study. So ethically, you should pay attention to that. So when it's very close to the scheduled end of a trial, the boards don't like to stop early because that means, we have to, if it's 1 month before or 2 months before or 4 months before, 4 months is not going to make a big difference unless a lot of things event rate is very high. So 2 months, 3 months, they don't like to do it. So that's why they only go up to 75. They don't go to 90%. Exactly. So, yes.
Interim analysis to monitor early efficacy and adverse events.
Okay.
So if there are adverse events, you have a separate mechanism to monitor.
Yes. So I will talk now about that's a good question because all I've been talking about is the efficacy part. We also have other things, which is the adverse events, and how do we monitor that? How do we incorporate that into this? So I'll get to that in a second. All right. So, and this gets to it a little bit. Arriving at the recommendation is complex. I've only been talking about the statistical considerations for interpreting the data on the efficacy, but that's not always a prescriptive role. Crossing the boundary doesn't force the Data Safety Monitoring Board to recommend early stopping. I'll say that again. Crossing the boundary doesn't force them to recommend early stopping. They take into account other factors other than the Z value. We statisticians think that we are God, and so statistics is important, but there are other things that are important too. So that is one element of the evidence that assists them in making decisions. It should never be the sole basis for stopping a trial, okay? Now, also these monitoring boundaries, I like to think of them as warning boundaries. There's, yes, warning, something is happening here. Pay attention to what's happening here. Look at safety. Look at adverse events. Look at secondary outcome. Look at sensitivity analysis. Look at subgroup analysis. Look at all of these other totality of evidence inside the study. look at the totality of evidence outside the study. That's why the members of the board are experts in the field who keep track of what's going on in renal disease, that keep track of what's going on in cardiology around the world. This is not the only study. So they don't base their recommendation on only one thing. They look at all of it. Now, how do they take into account safety? How do they take into account other things? The subjective things, who knows? Safety, as I said before, they look at it simple. But now they're trying to say, oh, yeah, we've crossed the boundary and it's substantial, but there's a lot of bleeding. Oh, there's a lot of people that are having atrial fibrillation. What should be recommending? Now they use their clinical hat on and they're thinking about, will practice change if I do this because I'll have to report the adverse events? Or should I give it more time for the adverse events to sort of settle down? Am I putting in danger the people by not stopping the study? Am I keeping information away from people outside the study? Who knows what they discuss, how they discuss it? Dr. X may say, no, 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 we need to stop. And Dr. Y would say, no, 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 we need to continue. That's why the minutes say, if they don't stop, they will say, we looked carefully at all the data presented to us and we do not see reasons why the study should not continue as per protocol. They don't say Dr. X said this, Dr. Y said that. As Kumar said yesterday, in the United States in particular, and maybe in Canada also, the minutes are of a NIH-funded study. Minutes of all discussions become public documents afterwards. And if I'm the widow of the guy that died, I'm going to read that and I say, Dr. X said not stop, I'm suing Dr. X. That's why those minutes don't say anything of who said what. They may keep other minutes, notes, private notes that they keep themselves so they remember what happened at the previous meeting, but they do not have official formal minutes just that reason, because they don't want to be liable for things. Okay, but how do we do safety? So before I get there, so all of these were group sequential designs. Even the PHRI one is group sequential design. And remember, those required that you say how many looks you're going to do, when you're going to do it, and that you have equal amount of information or events. The PHRI one actually does, but not all the other ones. People wanted flexibility. And in 1983 came the crucial paper that brought that flexibility, because you now not had to pre-specify the looks or the timing of the looks or the information size of that. Because these folks proposed, think of that 0.05 as money. You have 0.05 to spend, and you have as many looks as you want to do, but before you do that look, you say how much of your money you're going to spend. If you spend it all in the first look, then you cannot do any more looks. So for example, if I have 0.05, okay, and I want to look at time T1, T2, and T3. Before I get there, I have to say I want to spend so much. So if I spend 0.3, my critical value for 0.3 is going to be, I don't know what, 2.6. I'm just making that up. Now I have 0.2 left. So if I have 0.2 left, how much do I want to spend here? Well, maybe I spend 0.1. If I do that, that is, I don't know, 3.5. I'm left with 0.1. So I can, at the end, I have only 3.5. I have to have a Z that's big. So what do people want to do? Don't spend much here. Keep it for the end. So the PHRI boundary spends 0.00001 here, and then spends 0.00014 here. So at the end, it has almost 0.0499 9 there. That's why you get almost at 1.96. So you save it towards the end. And that's the notion that Len Demetz did. And they said, yeah, you can do it for calculating the O'Brien-Fleming, you can do it for Pocock, and we showed in our paper you can do it for the PHRI boundary. Now, all of these things have an impact on sample size, and people often ignore this. That if I use Pocock and I have 5 looks, I have to inflate my sample size by 23%. That's why nobody uses Pocock. If I do Wang-Sciatis, O'Brien-Fleming, it's 3%, or the worst case, if I do a lot of looks, it's 4%. So most papers will say, we adjusted the sample size for interim analysis by 4% because of this result here. Wang-Sciatis is somewhere in between. Hay-Biddle-Pito is 3%. We haven't done the simulations yet on this. We just did it for the alpha. We have to do it for the impact on the sample size. We have a graduate student here who wants to get a paper out. Welcome. We can work on that. Now, that's one approach. Group sequential analysis is the most commonly used approach for interim analysis. It is change your boundary by spending your alpha differently as you go along. I would say 99.5% of all trials that do interim analysis use this. Of those, I would say total 95% use O'Brien-Fleming, maybe 1%, 2% use Hay-Biddle-Pito, 1% may use Pocock, and we use the PHRI boundary, and people that work with us use the PHRI boundary. But there are other approaches to this. One of those is methods, that's the group sequential, but the other one is conditional power. This is a Bayesian type approach. It says, when I started the study, I designed it for 80% power or 90% power. When I start the study, actually power is 0.05, the level of significance. As I accumulate data, I can now calculate power. Power, you saw yesterday from Professor Jayasilan, is a probability. So I'm estimating a probability. I use the information I had it, whatever I had before is my prior. Now I have data, I update that, and I get a posterior probability. That's the Bayesian thinking. And if that power is small, I want to project what would it be at the end. If it's 0.5 or less, I may not even continue. It's futile to continue. So they call it stochastic curtailment or conditional power. Condition on the power that I have now. What is the power that I'll have at the end? So there's methods that are proposed as conditional power ones. There's these two guys that proposed, instead of doing repeated testing, let me do it with confidence intervals, repeated confidence intervals. I have never seen anybody use it except them. So, but I mentioned that it exists. And then there's another type of Bayesian approach that's not based on conditional power that was proposed by Spiegel-Halter in '94. Again, I have not seen it used. Some examples of how these were used. Famous one is the beta blocker heart attack trial that was stopped early in October 1981. This was a study that was planned for 48 months duration, close to 4,000 people getting propranolol or placebo. This is people that have already had a heart attack to not get a second heart attack. And they published the result, and they said they got a Z value of 2.82. People started reading it and said, whoa, 2.82 is close to 3. Didn't you know this earlier? October 81, why this doesn't happen to jump overnight. So they actually wrote a paper to defend themselves, and it appeared in December 84, where they said, yes, we were monitoring the study early. We crossed the 1.96 at October of 79, and 2 years later, we stopped the study. 2 years. people getting heart attacks, dying from heart attacks because they're on placebo. But no, we were monitoring. We only stopped it when we crossed the O'Brien-Fleming boundary because that's what we were using. But when did the O'Brien-Fleming boundary get published? 1979. And a new statistical technique is published. When is it adopted? Not immediately. So before the study was started, it didn't exist. They adopted it in the middle. And they adopted it and used it as a guide. But this is one of the famous studies that was stopped for it. The other famous study is the Women's Health Initiative that had a boundary for benefit and a boundary for safety. So this was given postmenopausal estrogen, and they wanted to see if it would be beneficial in preventing coronary heart disease, or it also caused the adverse effect of breast cancer. And the global index for benefit was here, but the log rank for breast cancer crossed the boundary, and they stopped the study early. No postmenopausal estrogen because it causes breast cancer. The more recent study that was stopped early was the COMPASS trial, and this is in patients with evidence of significant vascular disease. They were given an anticoagulant, rivaroxaban, plus aspirin versus aspirin alone, and they had the composite of MACE. It was a multi-center global study, and they used the PHRI boundary. And so here's the schematic of the PHRI boundary. The actual value was in November of 2016. They got 4.08. So they crossed the boundary. What did the DSMB said? Let's look at it in 90 days or so. So they looked at it in 90 days in January of 2017, and they were even higher, 4.59. They stopped the study at that point. So you can stop the study if you have substantial effects on that. And it was interesting because I joined PHRI in January of 2017, and a week after I was there, I had to deal with this. But it was an exciting moment at the time. Published the paper in the New England Journal, and also it was, I got the head statistician of the study to talk about how you do the monitoring. So the paper on monitoring emergent data explains how they did all of this part of it. Okay. Other considerations that impact the recommendation for possible early stopping is you don't only look at the current interim analysis, you look at consistency. If you had used the Pocock boundary, if you had used the O'Brien-Fleming boundary, if you had done this, you know, when we statisticians tell investigators, you have your SAP, stick to it, don't start doing other things. But this is exactly what the Data Safety Monitoring Board does. They want to reassure themselves that the result is something that I would be confident is true. So maybe because we chose one boundary, we got the result. But what if we had chosen another boundary? They look at consistency there. They look at consistency with previous analysis. They look at consistency with secondary events. They look at external consistency with other studies. They look at, despite the intent to treat, let's look at it in those properly randomized, so modified intention to treat. Look at it in the people that adhered. Do we get the same result? Do we look at adjusting for baseline imbalances because it was a small study? Let's look at that. So they torture the data, but none of those analyses are reported. These are done for them to reassure themselves that their recommendation is a good one. Who does this analysis? That poor unblinded statistician like Kumar that's sitting in the closed session, who is the only person besides the board members that knows what's happening, is being told, we want this analysis, we want this analysis, we want this analysis, we want this analysis. When do you want it? Yesterday. And when does he sleep? Who knows? He goes home and he's working 24 hours a day for 4 or 5 days getting these things to these folks. With the experience that we have, we know these things happen, and he knows this is going to happen. He already has these things programmed. So he prepares for the eventuality that they say this, because he knows that this may happen. That's why he hides at home, works from home, doesn't tell his wife. He says, what is the wife doing? No, I'm just working the stuff. Doesn't say what it's about, because he knows it's going to be that intense, and it usually is. And then despite multiple comparisons, they look at all the subgroups of interest, unadjusted and this and that. They really torture the data. Now, I already mentioned the statistical, we should stop the study early, like they stopped it at that first look with 50%. What happens? I don't get all the events I wanted. My confidence intervals are going to be wider than what I had expected to get. So you have increased variability. If you have wider confidence intervals, people will say, what's wrong with the study? There's too much uncertainty in your estimates here. So you have reduced credibility. You may be biased at this random high. That's why we do the 90-day thing. We don't get those false positives. But this is what most investigators worry about. Oh, I really wanted to know what is the long-term effect of this intervention. And we've stopped it early. We're not going to get those long-term effects that we had planned on studying. Like if you're looking at that neurodevelopmental one and you stop it, if your outcome is at 12 months, you still may be interested on 24 months, but now you don't have enough people that got to 24 months to study it, right? So that's what you sacrifice. You sacrifice secondary events or long-term events may be affected. But you do it because of ethics. You have to do it. So when do you do interim analysis? As I said, the study starts from an idea to funding. You start the trial, you end recruitment, you end follow-up, you do analysis and funding. The meeting of the boards may start sometime. Usually there's one here before this trial is started. That was that review of the charter, et cetera. But the first meetings are looking at safety. They're looking at recruitment. They're looking at follow-up completion and adherence, et cetera. All of them will look at that. But now if you recruited, then they stop looking at recruitment. They continue looking at safety. They continue looking at follow-up. And then at some point they say, okay, let's start looking at efficacy. And then they look at safety and efficacy and futility, et cetera. And they keep looking at all of the other stuff as well. So the purpose of the meetings changes as you go along this. So I'll stop here.